Our first speaker will be Sacha Gnjatic from the Human Immune Monitoring Center. Sacha received his PhD in immunology from the University of Paris and later completed a postdoctoral fellowship at Memorial Sloan Kettering here in New York City, where he later stayed for some time before Mount Sinai was fortunate enough to recruit him in 2013. He joined the faculty here in 2013 and is a part of the Tisch Cancer Institute as well as the Immunology Institute. He also serves as Associate Director of the Human Immune Monitoring Center. His research focuses on human immune responses to cancer in an antigen-specific manner. He also looks at characterization of serologic and cellular immune responses against tumor antigens such as MAGE-A3 and NURIC-ESO1. And he's interested in studying immunomodulatory markers and their relationship to tissue architecture and antigen presentation. Today he's going to be introducing to us a technique that was developed in his lab using multiplexed immunohistochemistry to study the tumor microenvironment. It's my pleasure to welcome my mentor, Sacha Gnjatic.
Thank you. Thanks, Ravi. And also I wanted to thank Laura, Christy, and all the people from the Immune Monitoring Center who helped organize the meeting. So as Ravi said, we're switching now to, after hearing all these amazing talks about single-cell methodology, to how do they map onto the lesion of a tumor or a microenvironment within the architecture of a tissue. And these are disclosures that I usually present, but none of them are relevant to this talk. And I wanted to put this in a broader context. We've been interested in the Immune Monitoring Center at looking at tissue lesions in every potential dimension to answer questions in particular in my own interest related to cancer immunotherapy, where we know that so many of the new drugs are now standard of care based on the immune system, and yet we know also that only a fraction of these patients respond to these drugs, and we don't truly know why those patients respond. So we here in the monitoring center, as well as through now a newly funded effort called the CMAC, we are asking those questions by doing these high-dimensional approaches, both from immune profiling of the tissues and the interrogation of the functions, immunopathology, which I'll talk today about, immunogenomics and microbiome analysis, and all this needs to be supported by a huge bioinformatics effort. So really, this is part of a larger group, but immunopathology is in itself interesting, and I'll go through the rationale why we think it's important to characterize the cells, not only for the composition within the tissue, but also for their organization within those tissues. And give you explain to you the method that we've developed in-house from multiplexed immunohistochemistry called MIX, and a few examples of applications, and also how we're helping analyze this platform. So there's a lot of literature now out there, if you look in across the things that have been published, showing that having immune cells, in particular T cells, infiltrating tumors is a good prognosis for a majority of different tumor types. And so in this meta-analysis, they saw that almost every paper published out there looking at T cell infiltration was associated with a good prognosis. And it's been even proposed in the context of colorectal cancer to be better at distinguishing patients with good and bad prognosis than the tumor node metastasis classification, where you can now reclassify patients based on low infiltration as having as bad of an outcome as stage 4 patients, and vice versa. These early stage patients, the advanced patients could do as well as early stage patients if they have a high infiltration, in particular both inside the tumor and at the invasive margin. And besides also being shown for being of prognostic value, these T cells are also of importance in terms of their predictive value to immunotherapy, in particular to these new checkpoints like the PD-1 blockade, for example, in melanoma, here a drug called pembrolizumab, where patients who responded were almost all of them the ones who not only had more T cells at baseline, both inside the tumor at its invasive margin, but also recruited more of them during treatment. However, I don't want to mislead you by saying that only T cells are important, and there are some counterexamples. There's this paper that was published in renal cell cancer showing that actually T cells are associated with just higher grade of disease and are of poorer prognostic value, surprisingly, while conversely there are more NK cells in this disease which are associated with better outcome. And in lung cancer, even though T cells are very important prognostically, there's also evidence that other cells could be even better potentially, such as follicular B cells and dendritic cells, both at early and late stage, where having a lot of them is associated with a good outcome. And then there's also not just the density of these cells, but what's their functionality and markers they express. And so one of the most common markers in the field of immunotherapy with checkpoints has been PD-L1. And early on, it's been proposed that PD-L1 is important as a potential prognostic marker, predictive marker of response to the checkpoints, but not necessarily just by its presence, but where is it present on. And in this particular study, they looked at PD-L1 on immune cells, in particular macrophages, and saw that these were the ones that were actually the most important for being able to predict outcomes. So we're looking at all these potential markers, but how can we assess them all comprehensively, given usually the small amount of tissue you obtain from these patients that have tumors, usually from biopsies, and that do not allow you to do really all these markers consecutively by standard immunohistochemistry. So this is really a problem of sample availability in general. And also to do the multiplex methods that are commercially available is typically quite expensive. And so we were thinking, can we democratize access to multiplex immunohistochemistry in some way? And what would we need to do that? So the idea is to use the simple method that already exists in most pathology labs, which are chromogen based and include standard flow that's for any single marker, and use also paraffin embedded tissue formalin fixed, which is really what's mostly available in archives, and also that's usually where tissues go first when you obtain them from a biopsy or surgery. And we know also that most labs do have access to either macroscope and more to slide scanners, where digital pathology is becoming more and more of the norm. And again, it has to be affordable. So this is the idea was to develop this low-cost and sensitive method that uses standard pathology methods to allow for the staining of consecutive markers on these FFPE slides, and to look not just at individual markers, but also potentially their colocalization, and that would not have the issue of cross-reactivity or autofluorescence or photo. So there was a very talented postdoc who worked in my lab, Romain Remarque, who was also co-mentored with Miriam Murad. And he thought of simply developing a method which would reuse a single slide over and over using standard chromogen labeling. So the first steps are identical to what would be done in a normal pathology lab, which is to take the slide, deparaffinize it, and then stain it after an antigen retrieval step. So this is quite important in FFPE, that you have to basically make the epitope accessible to the antibody, and that includes boiling and putting specific pH conditions. And then you put your primary antibody, your secondary, which is linked to HRP enzyme, and then chromogen revelation, and scan the slide. And so you can get an image like this. This is a red chromogen called AC, which gives you the CD20 B cell follicles in the lymph node. And then you can remove the cover slip, perform a dehydration. And the trick here was to use this dye, which is actually labile and can be removed by simple chemical destaining, where you remove mostly the signal, and then you can either keep that slide away for a while and just can come back to it later, or go into the next step where you now do a blocking to prevent any cross-reactivity from the first antibody step that you've done, but also a new antigen retrieval step, which will remove a lot of the primary antibodies, though not completely, that was originally stained there, and also be for the next marker optimized conditions for the staining. And you can do that now and see CD3 staining, for example, here, and repeat that. We saw maybe up to 10 times or potentially more until you start potentially getting some tissue degradation. So this consecutive method is just here an example in a small bowel neuroendocrine tumor. Can be adapted to a whole tissue, which is really quite nice. So you can zoom in areas here that have lymph node structures, lymphoid structures like germinal centers with B cells on the right, T cells around them on the left, as well as some macrophages. And it's a single cell resolution, so you can really zoom in and see the heterogeneity from area within the tissue here, where you see that not all these cells are equally densely expressed everywhere. Some of them sit in the periphery of these tumor tissues, and other are a bit denser, so you can go then across tissue on the other side of the muscle here and see that fewer in this case, and eventually quantify them for expression. Well, I'll go rapidly into just some of the technical validation to make sure that the method works well. In the colorectal tissue here, where we have some tumor areas and stromal areas. And so first, the first step that was stained here was CD2, which is a T-cell marker shown in brown here. And then the next step that I'll show you were artificially colored just to be able to visualize them better. So we co-stained, and we stained after the next step with CD3 and overlaid the image. And you can see that all the CD3 cells are also CD2 positive, which we expected, both cell markers, as well as you can then stain for CD8, which is only on a subset of the CD2, CD3 cells. And indeed, we can see now that some of them are indeed co-stained, but there are 3 markers now on the same cell without these hindrance, spherical hindrance. You can then go and put like a CD20 marker and make sure that these cells are not T cells, and indeed they're different, so you don't see any overlap between the cells. You can add additional markers such as DC lamp for dendritic cells, and even see some interaction between T cell and dendritic cells. Look at tumor antigen, MAGE3, which in this particular tumor was negative, so that's also kind of a nice control to see that not everything binds. Ki67, which is a marker of proliferation. This one you can detect both in the tumor cells as well as the immune cells. So some now of the tumor cells light up, but also some of the T cells that are proliferating. And then add another marker like FOXP3 to look at which T cells are actually regulatory cells. So now we have 4 markers on the same cell potentially. 3 FOXP3. So they shouldn't be FOXP3 and CD8 together. And so what I want to emphasize here is that within this method, we used several clones that had the same isotype and from the same species, and yet we didn't see any problem of cross-reactivity or carryover. And this we confirmed also through permutation of staining order experiments, where we took adjacent slides and stained them either in the order shown here, or did permutation where we stained first lamp and then CD2, CD3, and quantified each marker, and whether it was the first staining done or after 7 previous stain, we don't see any loss of antigenicity happening, and they're basically the same density, at least for these constitutive markers that are expressed on lineage cells. We also see the same for CD3, where we can restain and not lose really that much intensity of signal, and so basically every time there are new epitopes available when you redo these consecutive method. And we keep also the tissue integrity even for these fragile lung tissues, for example, which even after 9 cycles we can still detect these. So then next was how to analyze this tissue, and this is recent work from Guray Aktor, a pathologist who is now working with the Immune Monitoring Center, where we try to register images. So on the left are the original images that were copied, and on the right were after registration. So you'll see here on the left that it moves every time from image to image, and on the right it stays more stable. So you're now able to really make sure that you can co-express those data, and when you segment the cells later on, quantify each marker on the same cell rather than to guess which cell was which. That helps a lot. And that allows you then to deconvolute this picture and take the channel corresponding to your stain and add it to your images in an overlaid fashion. And to visualize it more easily, and to be able to also quantify these images, we then convert the images into a pseudo-fluorescent staining type. So this is blue showing hematoxylin actually originally stained, which is now looking like a DAPI in a way as a fluorescent, showing you where the cells were. And then you can turn on and off the individual layers, such as in this case CD3, CD8, seeing that they're expressed, CD20 and cytokeratin, they're not, and macrophages, and some of them are CD6. So you can look on and off and quantify them individually or turn them all on and see now more of the complexity in how they organize such as cells in specific clusters. So that can be applied now to actual clinical questions, and this you may have seen me present already, but it's a nice picture of a melanoma lesions of a patient treated by Jed Walchuk and Taha Mergub at MSKCC. The patient overall responded to ipilimumab treatment after blockade therapy, but this particular lesion persisted, and they removed it and asked us to look for what's happening in this particular tumor. And the tumor expressed PD-L1 itself, but also had a lot of macrophages that formed a ring around that particular PD-L1 positive tumor. And when we looked at mature dendritic cells, they were there, but mostly outside of the tumor itself. There were tons of B cells self-organizing again as germinal centers, and many, many T cells, but again, all excluded from the actual tumor that was PD-L1 positive. And some of those T cells were FOXP3. So now you can get a better idea that even though this tumor looks like it has a lot of T cells overall, they're just potentially explaining why the particular tumor is still going on, because they weren't getting right where they were supposed to. So we're also increasing the number of markers that we can analyze. One of the good things about this method is it's pretty much mix and match. So you can take the antibodies of your choice and don't have to follow a specific pattern of order of staining or a specific number of markers that would work together well. Here we have developed now panels for dendritic cell subset, macrophages, and T cell profiling, and we continue to add. And it's not just for tumors, so this is a collaboration with Dirk Homan, who has now implemented the method here at Sinai with Verena van der Heide in lesions from diabetic patients, and they could look at other markers such as insulin and glucagon, and it works as well. We can even keep specific markers as reference using a second chromogen that is not labeled with the method, and so in brown here are cytokeratins that stay throughout as a sort of specific marker of tumor cells, and then you can stain on and off with the other markers of immune cells around it. And we even applied this to bone marrow, even though this is still preliminary, but it looks like even in these decalcified bone marrow tissue, we're able to see here the myeloma cells on the left, and which tumor antigens they may express, like MHC1 and MHA, and heterogeneity at a single cell level of this expression. So the final steps are image analysis. So how, now that we saw we can look at these cells, visualize them, and look at co-expression, how do we quantify them? And again, with the help of Gurai, we took advantage of an open source software called QPath that was developed from the UK, and which is really remarkable at segmenting cells, and it's free, which is quite amazing. And so you can now be able to look at the whole tissue and segment the cells, and even then quantify them by their markers. So in red are the stained cells, in blue are the remaining segmented cells. And through this collaboration that I mentioned in the beginning of my talk with the NCI grant, we're now comparing our quantification with two other sites, MD Anderson and Dana-Farber. And you can see on the right that our quantification of the same tissues at different sites using different algorithms and different machines are still pretty remarkably similar. So that's a good idea for us to be confident in the reproducibility of this data. So you can now apply this method to tissue microarrays, which is a nice application of this mix, because with one slide or several slides of the tissue microarray, you get tons of information from many different patients, and you can use the software to automatically find each individual patient core within the microarray, and then segment them, quantify them. And we applied that first to a lung tissue microarray that was commercially available of only 72 patients. And we could confirm some of the literature showing CD3 cells are, for example, predictive prognostic of outcome. If you have a high CD3 count, you're doing better on overall survival, but also propose potential novel combinations of markers that could be even more predictive, such as a high dendritic cell and low neutrophil ratio being the best predictive marker in this particular cohort. This is also unpublished data in another microarray of head and neck cancer patients, of about 500 of them, where we looked at CD3 infiltration and also saw that it was a good prognostic factor in this particular patient, in particular also within the lymph node metastasis, but not so much in the normal adjacent tissue. And we could look at B cells and find that these were associated actually with the T cells. So the more B cells you have, all the B cell positive patients were also T cell positive. And as a result, you didn't really improve as much on the survival prediction. They were not really helping separate the curves that much more. But you can also look then at associated with clinical outcomes and clinical parameters such as HPV status. We saw that HPV positive patients had more T cells in them, while the smokers had fewer T cells, interestingly. And as a result of the higher threshold of T cells in the HPV population, these were actually no longer predictive using the median value, while they still were in the HPV negative. And you can then make combination with smoking status as well as HPV status and see that the best prediction is actually in patients who are smokers but not too much and have HPV positive, HPV negative and high CD3. So within the non-smoker, actually, there was not as much of a separation of the CD3 count. Maybe hinting at potential antigens being recognized, which we try to address by the presence of MAGE antigen in these patients. And surprisingly, for MAGE, which is a known cancer-testis antigen expressed in different tumors, including head and neck, we didn't see an association with more T cell in the presence of more antigen. However, we knew that MAGE expression itself is a poor prognostic factor, and it is additively worse if you now compare it with CD3 together with MAGE. You can see that the patients who had a high CD3 and low MAGE were the best patients, and vice versa. High MAGE, low CD3 did poorly. And the final few slides I want to show you is how do we now also go further and use some of the deep learning algorithms that also are available through the QPath software to not only quantify these images, but characterize them in an automated fashion to try to see, can we see general patterns emerging within these tissues? And so here is just a way to show that we can train the system for a specific cell type, and based on 67 different features found by QPath, such as circularity, cell-to-nucleus area, and even markers, you can automatically then assign this particular cell type to a tissue or to basically give it an annotation, such as whether it's tumor, normal, airway, etc. So this is an example, again, in a pancreatic neuroendocrine tumor now, where you can zoom in at a very high level in the CD3 stained cells here, and then apply this deep learning and see the different colors now assigned to each particular cell. In red, the tumor, in orange are the normal tissue, which are very little here, except in an area when we zoom out, and green stromal areas, and in purple are cells that look like they're immune cells simply based on their shape. And so you can now start thinking about things such as where are the tumors in relation to other cells, and look at nearest neighbor analysis, such as are the T cells really in close proximity to tumor cells or are they close proximity to other cells. And in order to do these things, you have to somehow convert these images into data, because you cannot really quantify them one by one. And so this is something we started doing now with Adib and again Gurai, where from these original images that we propose as fluorescence, you can derive counts and individual values of both XY coordinates and each individual markers, and then put them in the same pipeline that are used for dimensionality reduction, such as CyTOF or even single cell, where you can make some of those famous Disney graphs and see where those cells cluster together. So first you can recreate digitally the individual original image by their markers and their XY coordinate, but then cluster them, and here it's showing where the CD68 positive cells look on this Disney map, and each dot being a cell, and CD20 cells in there, and then based on this cluster, quantify them and see, for example, co-expression of some markers that I showed you earlier, like CD68 cells having a subset of CD206 and 206 positive and 206 negative. Same thing for the CD3, where we can infer the CD4 cells as the one being CD8 negative. So this is still work in progress, but we're hopeful that now we can maybe solve the issue that are still remaining, such as potential false positives, where you have some co-expression that make no sense, which could be due to artifacts of the segmentation itself and how we're basically quantifying our tissues. And so this is now a method that we propose to apply as part of a larger, as I said, effort for immune monitoring of tissues. And that could also be complementary to some of the talks we'll hear next about tissue imaging at even much higher dimensional levels, such as the MIDI or other metal-based like tissue CyTOF as well. And this is part of like basically effort where we're trying to go from the whole tumor characterization by radiomics feature to the tissue analysis, the organization, then to the single cell analysis, and all the way to the genomics. And finally, this we're already starting to integrate in some ways by doing these deep tissue analysis with IHC here and quantifying the various areas in lung tissue, then taking one area of the same sample and putting it through MIBI in collaboration with Stanford here that did a much deeper 35 marker analysis with the same tissue as a dissociated cell through CyTOF, and then analyze it by single cell, and try to now relate each one of those, not only for the genomic profile, their proteomic profile, but also their localization in tissues. So I hope I convinced you that there is a need for these high-dimensional monitoring techniques, in particular the tissue-based one, multiplexing. This is a method that we hope will be used also more broadly because it's pretty easy to implement. It doesn't require much besides a slide scanner at this point if you want to do it manually, although we're in the process of also automating it so that it can have a higher throughput. And it's really hopefully going to work better as we go along for these image registration and analysis, thanks to the deep learning tools that are being developed by the Immune Monitoring Center here. So I want to thank again Romain Remarque, who developed the method, and Gurai, who helped a lot in the image analysis, and Ilaria, who made a lot of these things as well, and Robbie, who's now going to start doing more of these analyses, especially comparing this to MIBI, and then all my clinical collaborators who contributed to also some of the samples that I've shown. So, thank you.